Then you have a snapdragon that is white. And basically what we already know about this process is that usually there's a recessive allele and a dominant allele. But in the case of incomplete dominance, when you cross-pollinate these two varieties, instead of getting a red, 3 reds to 1 white, which is what we studied in our last lecture, instead you get a pink. Hence it's called incomplete dominance. And this just really refers to the phenotype that reflects the amount of a gene product from an allele. Okay. The next complex pattern of inheritance involves a process called codominance. And this brings me back to our first slide. To me, there is no such thing as incomplete dominance. Okay? We each receive 2 alleles for each gene. We each receive 2 alleles for each gene. Both alleles are being expressed. Both alleles are being expressed. Both of them are producing protein products. So in effect, the concept of incomplete dominance is what I would call sort of inaccurate. It's an extreme inaccuracy. Incomplete dominance just basically is referring back to the idea that both alleles, one allele from each parent, for each gene that we have, both alleles are being expressed, both alleles are producing protein products. So in effect, all of our genes are actually codominant. Both alleles, all of our genes are in truth codominant. Because again, for every gene we have, we get an allele from each parent. And both of those alleles are being expressed. They're both producing protein products. Therefore, we can consider all genes to basically be codominant in that you have protein products expressed from both alleles. Okay? The concept of dominance really refers to phenotype. It refers to phenotype. Codominance, and this is where your book gets a little bit light on this subject. Codominance is referring more to the molecular interactions of the protein product produced by each allele. So I'm going to say that again. Codominance actually is referring more to the function of the protein products produced from each allele for every gene that we have. Are we okay? Can I repeat that again? I'm going to repeat it again because, again, your book really doesn't go into this very much. It doesn't really go into it at all. But all of our genes are examples of what we would call codominance because each allele is encoding a protein product that is going to have a biochemical function. Each allele is encoding a protein product that's going to have a biochemical function. When we think in terms of phenotype, yes, sometimes the biochemical product of one allele might have a stronger effect on the cell than the product from the other allele. That's when we think in terms of phenotypic dominance versus phenotypic recessive. But in effect, all of our genes have 2 alleles, and both of those alleles are actually producing protein product and having a biochemical effect on the cell. Do we understand that concept? Okay, this is very important. And in your study guide, I wrote a description of this process so that you can understand really what's happening at the molecular level. Okay? All right, so at the molecular level, at the molecular level, one of our best examples is the ABO blood groups. The ABO blood groups. The ABO blood groups actually refer to a glycoprotein that's on the surface of our red blood cells. That's what the ABO blood groups are referring to. It's referring to a glycoprotein that's on the surface of our red blood cells. And the glycoprotein A, this is actually, glycoprotein A is encoded. It's encoded by a glycosyltransferase. We have a gene that encodes an enzyme called the glycosyltransferase. And the gene, the enzyme that is the glycosyltransferase for blood type A actually attaches an N-acetylgalactosamine to the surface of the red blood cell. Okay? So, the glycosyltransferase, encoded by the enzyme that gives you the A blood type, that glycosyltransferase actually transfers an N-acetylgalactosamine to the surface of the red blood cell. In the case of the B blood group, what you have is an enzyme. It's an enzyme that actually attaches a D-galactose to the surface of the red blood cell. And in the case of the O blood group, what you have is a gene that encodes a non-functional transferase, glycosyltransferase. So that's the difference. That's the biochemical difference between the A, the B, and the O is the glycosyltransferase that's actually putting carbohydrate chains onto the surface of our red blood cells. So the A carbohydrate chain is different from the B, and that's because there is a form of enzyme that adds the A-type carbohydrate chain, and an enzyme, a different enzyme, adds the B-type carbohydrate chain, which is, like I said, D-galactose. Okay? And the O blood group, that just means that these individuals have no functional enzyme. Okay? There's really not any real strong known biochemical function for these carbohydrates on the surface of our red blood cells. And just for also for you to understand, not only are these carbohydrate chains on the surface of our red blood cells, they're on a great many cells throughout your body. So it's not just the blood cells, it's also on all of your epithelial cells, it's on the cells that make up your blood vessels called the endothelial cells. All of these have these specific carbohydrate chains. Okay? All right. So these are the alleles. The glycosyltransferase gene A, glycosyltransferase gene B, and the non-functional glycosyltransferase. That's what we have. So this is the basic biochemistry that I just described to you. Now we're going to go into more of the genetics. Since we have, for each of these enzymes, we have an allele from our mother and an allele from our father. And so for somebody who has the A blood group, that means that their phenotype is the A blood group, but their genotype, they could be homozygous for the allele that encodes the A-type glycosyltransferase, or they could be heterozygous, where they have one allele encoding the A-type glycosyltransferase, and then the other allele would encode the non-functional enzyme. Does that make sense to you? What I would also stress is that for individuals who are of blood type A, if they're this heterozygote, does that mean that they probably have about half the number of carbohydrate chains on their red blood cell surface? Yeah, sure. Absolutely. Absolutely. They only have one allele's worth, okay? Whereas if you have, your genotype has two alleles, both encoding the carbohydrate chain, then your red blood cells are going to have that many more of these A-type carbohydrate chains on the cell surface. Okay? Can I explain anything further, or is this reasonable? Is this reasonable to understand? Okay. All right. So, similar situation with B blood group. The B blood group, an individual would be homozygous for two alleles, both encoding the glycosyltransferase that transfers the B-type carbohydrate chain to the surface of the red blood cell. Or this individual would have the allele encoding the B chain, but then also the non-functional enzyme. So the difference between this and this and this and this is the amount of the carbohydrate chain on the red blood cell surface. Does that make sense to everybody? Anybody have a question? Or can I repeat anything again? None of this is in your book. Okay, the question is this an antigen or an antibody? It's an antigen. It's called an antigen, yeah. It's called an antigen. And that's because if you transfuse an individual with A-type blood into an O-type blood person, what's going to happen? The immune system of the O-type person goes nuts. It can't deal with this, the blood will clot, and your patient will die. Okay? That's why it's called an antigen, because it induces an immune response. It induces an immune response. So this is the A-type antigen, this is the B-type antigen. Individuals who have O blood types don't have any of that. Okay. So then we have AB blood type. AB blood type means that you have the allele encoding the glycosyltransferase adding the A-type carbohydrates to the cell surface. And you also have the allele encoding the B-type glycosyltransferase adding the B chain to the cell surface. And again, somebody who is type O doesn't have the functional glycosyltransferase, so you will not find any of these carbohydrate chains on the surface of the red blood cell. Okay? So do you understand now the concept of the ABO blood groups? Okay. All right. So if you had to figure out, based on what I just told you about the effects of the immune system on transfusions, which blood type would be what you would call the universal donor? This one. Oh, absolutely. This is the universal donor because anybody can get this blood, right? Anybody can get this because there are no A carbohydrate chains or B carbohydrate chains to elicit an immune response. What is the universal acceptor? AB. Absolutely. Okay, so you know this. You're good. Okay, it's just logic. It's just logical, right? Okay. Does anybody have a question? Any questions about question? The question is, which is most recessive? They're codominant. There's no recessive here. There's no recessive. What you're going to see is an individual is A, or B, or AB, or nothing. Okay? When there's nothing, I guess you could say, in truth, that's the most recessive, because there's nothing at all. Okay? All right. Question? So, who wants to play Who's the Father? Yeah. This is one of the ways to determine parenthood. So for example, if you have a mom who is type A, and you have a dad that's type O, and the baby is AB, what do you think? That's not the dad, right? You know how often that happens? At least 10% of the time. At least 10% of the time. That's just based on blood groups. If you were to actually look at genetic sequence, then probably it would go up. Probably 15 to 20% of the time. Okay? But what? Dad's job was done 9 months ago. Right? And so we always know who the mother is. But you don't necessarily, you cannot count on the biological father. Okay? You never can. And so this is what happens. See all these nice little black puppies here? Well, that's the kind of thing that goes on. That's the kind of thing that goes on with people too. Okay. All right. Does anybody have a question? Okay. All right. So now we understand the idea of codominance and how in truth all of our alleles for all of our genes are codominant because they're producing protein products that have a biochemical effect on the cell. That's the most important thing I want to make sure you understand. Okay. Then we go into the portion of complex inheritance patterns where gene products will then start interacting with each other. And that's when we come into the concept of epistasis. Epistasis is a genetic inheritance pattern where one gene, the product of one gene, is going to alter the phenotypic expression of another gene. And a wonderful example of this is, I guess, dog coat color. That's what our book likes to use. We like dogs. I mean, look what it brings you. All kinds of interesting questions, right? Anyway, so what actually induces the dark color versus no color at all? An example of this would involve, for example, a transcription factor. Transcription factors are these proteins, if you recall, that bind to genes to allow them to be expressed. So a transcription factor that binds to a gene that activates pigment production. So the gene that's altering the expression would be the transcription factor. The transcription factor is a protein, and what that transcription factor does is it binds to a gene that is then going to encode either pigment or enzymes that generate pigment. And that's an example of epistasis, and this is the example that you see here. So, for example, if you have a lot of that transcription factor around, and if that transcription factor is binding to the gene that encodes an enzyme for making pigment, you're going to get a dark-coated dog. Whereas if you have very little of that transcription factor available, and that would be an example shown here with the lowercase b, and you actually have a gene that encodes a very, very sluggish enzyme involved in generating pigment, then you're going to get much lighter coated dog as a result. Okay? So these are examples of, this is an example of epistasis, where one gene product, and that would be, for example, a transcription factor, would alter the expression of another gene. And in the case of this, you can talk about a transcription factor that activates expression of an enzyme that's involved in pigment production. Okay? All right. Can I explain this any further? Question?
So what's the significance of epi in the...
Epi It's just called epistasis. Epi means outside, and stasis means continuation. And why they call this gene expression pattern an example of epistasis, I've never understood that. It doesn't make sense to me. Okay. All right, then we can have another form of complex genetic pattern that's called polygenic inheritance, where you actually have multiple genes that determine a single characteristic. And in this case, they were looking at known genes involved in human skin color. And for example, we have numerous enzymes that function in a pathway to generate melanin, the pigment molecule in our skin. So we have a number of enzymes that function in this process. And so, for example, the various enzymes could be labeled A, B, and C. And you could actually kind of determine just how light or how dark somebody's skin was going to be based on the efficiency of each one of these enzymes, whether it was very efficient or a little less efficient. Okay. Can I explain anything further? Okay. So very efficient, you get much more pigment production. Less efficient, you get less pigment production. And we've already known when we've studied enzymes how they can have variation in their ability to produce protein product, right? They do. That's one of the big sources of variety in all of our different biochemistries. It's just the efficiency of our various enzymes. Does anybody have a question? So this is called polygenic inheritance, when you have actually more than two genes that are going to determine a single phenotypic characteristic. Is how light or dark the skin is. We've also talked about epistasis, where you're looking at the effect of transcription factors, activating genes, and then we've just talked about polygenic inheritance. All right. So now I want to talk a little bit about Mendelian inheritance patterns. This isn't terribly Mendelian because you're looking at variations in biochemistry. You're looking at variations in amounts of protein that are affecting an overall phenotype. It's a little harder to determine versus purple flower color versus white flower color, right? Mendel primarily looked at traits that were easily identified and quantified. Okay, so you had to have a characteristic that could be immediately identified, and then you could quantify the numbers of offspring or individuals with that characteristic. And so we're going to look at some pedigrees, we're going to look at Mendelian inheritance patterns. And for example, this is called a widow's peak. A widow's peak versus a smooth hairline. And it's known that widow's peaks that establish this little point coming out here that this seems to be dominant. That if you have an allele encoding this, and it's the pattern of hair follicle formation in fetal development. So always there's a biochemical basis for all of this. So in the case of widow's peak formation, what you're looking at really is development of hair follicles out onto the forehead during fetal development. It's not life or death, right? No, not at all. But that's what this is. It comes back to a biochemical function, and it's known that if you have this pattern of hair follicle formation that it tends to be dominant over the smooth, rounded hairline. That's known. Why? Nobody knows, okay? But simple Mendelian genetics, it's a trait you can identify and you can actually quantify. So for example, in Mendelian genetics pedigrees, squares are male, circles are female. If something is filled in, if the square or circle are solidly colored, that means they have the trait that's being studied. So for example, in this pedigree right here, you have a dad that has a widow's peak. The mom had a straight hairline. They had 4 kids, 2 of them with widow's peaks, a girl and a boy, and 2 without. All right, so this young man with his widow's peak, just like his dad, marries a young woman, or at least has kids, with a young woman who also has a widow's peak, and they have 2 kids, one with a widow's peak. This girl has a widow's peak. This one does not. And we don't really know which genotype she has, do we? She could be homozygous, dominant, or heterozygous, dominant, right? Okay, that's the concept. That's the concept of Mendelian genetic pedigrees. So we understand now what the symbols mean. Circle female, square male. Filled in, it means that this individual has the trait that's being followed. When it's a clear circle or square, that means that this individual does not have that trait. Okay? All right, very similar situation with attached earlobes versus free-hanging earlobes. It's the same thing. The attached earlobe is recessive. Free-hanging is dominant. And so this is the example of a pedigree that you would get. So what you would have is the attached earlobe. The attached earlobe would be indicated here with this young man. He has the attached earlobe. He's homozygous recessive. The mom here, or the female, is heterozygous. She has an allele that encodes a free earlobe and an allele that encodes an attached. But because it turns out her earlobe is free, then this is the dominant phenotype. The dominant phenotype. And this is the kind of result that you would get from matings. Sometimes you always know the genotype when it's a recessive gene. You always know the genotype when a person has that particular recessive phenotype. But if a person has a dominant phenotype, that individual can be homozygous dominant or heterozygous with the dominant allele. Okay? We are all right with this? It's logic. Okay. Now I'm going to talk a little bit about genetic disorders. One of them is albino, the condition of albinism, where very little skin pigment is made. Does this have a significant medical effect on this child? As long as she stays out of the sun, no. Okay? She has to absolutely stay out of the sun. Your protection against damage to your DNA from ultraviolet rays of the sun relies upon melanin or pigment. And if she's not making any, then she has no protection. It's known that we have at least 9 genes. We have at least 9 genes that are actually involved in correcting damage to the DNA as a result of UV light from the sun. We have at least 9 genes that encode DNA repair enzymes that their only role is to actually repair damage to the DNA from UV light. Okay, that is. UV light is actually our most dangerous environmentally damaging